tell you guys more about our ICSA mentor-mentee program.
Yeah, thank you very much. And for a brief introduction, my name is Yuxi Liu. So I'm actually the chair for ICSA Mentor-Mentee program, and this is a newly founded program here at the request of many people, and some of the people here maybe submitted a request before, and we listen and we want to found this program trying to help more people here. So what this program is going to be about, because a lot of people are actually going to graduate or as their early career here. So for these people, for example, some of them may not have enough industry experience, and some of the people could actually experience a little bit culture shock when they switch from academia to industries here. And from this group here, and the ICSA wants to help better to understand your needs and trying to find more experienced and seasoned statisticians in industry, trying to provide some of the professional guidance for people's career development. And right now, we are trying to recruit both the mentees and mentors here. And the mentees, we are not only going to cover the graduate students, and also we want people who are like early career statisticians there and interested in industry roles here, and mentees here, and we want experienced professionals here from who has been working in the industry, for example, for quite some time, and with a passion to do the mentoring here. And in terms of the program structure, we have been launched is actually a 1 hour Zoom-based session, and each time we will have 2 to 3 mentors join, and we want to do this one rotate different industry sectors here. And different from most of the meetings you see, it is going to be highly interactive here. And you can even submit some of your questions ahead of the time, or in many situations there, you can ask the question directly to the mentors there in the meeting, so that other people can also benefit from your questions there. So we have a QR code here. So if you're interested to become a mentor, if you have enough experience there, feel free to register through this QR code here. Next slide. And this is actually our first meeting of the ICSA mentoring launch, and it will happen on October the 1st. And we actually are going to focus this time on pharmaceutical industry, and we have 3 really experienced people joining as the mentors here. You can see Mac, Wei, and also Xun. So they came from different pharmaceutical companies there. And also in this situation, if you take a look, all of them are at VP levels there. So, and especially Wei, he is the one in Lilly trying to do a lot of recruiting right now. So he will have a lot of experience to share. And if you're interested, and please scan our QR code here and try to register, and we will send out the link as soon as possible here. And also, we really appreciate the webinar of ICSA to give us opportunity to share this information. Thank you very much, and we hope to see everybody in the future. Yeah.
Okay, thank you, Yushi. And then I will continue with our webinar today. Let me do the next slide. So today's webinar is our wheel of analysis strategy on handling missing at random data at Estimand. And I will pass the time to Dr. Deli to introduce our speaker today.
Okay, thank you, Betty. My name is Deli Wang. I'm a co-chair of this webinar series. I will introduce today's speaker, Dr. Bidan Huang. Bidan is a senior director at AbbVie. She got her PhD degree from Tsinghua University in China in mathematics, and got her PhD in statistics from University of Illinois at Urbana-Champaign, UCIC. She is currently leading multiple market and early phase compounds in development in various disease areas, such as obesity, pulmonary disease, men and women's health, antiviral, anti-infectious diseases. Since she joined AbbVie, she has extensive experience in leading statistical efforts in advancing pipeline drugs from phase 2A to phase 4, as well as submission and post-marketing support in gastroenterology, cardiovascular, neuroscience, and immunology therapeutic areas. She's also a research fellow in every community of science. So that's all I want to talk with Bidan. She has a lot of experience. So Bidan, right now, floor is yours.
Thank you so much, Deli, for the nice introduction. And thank you for everyone who's coming to the webinar on a Friday afternoon. So now let me share my screen and share. Can you all see my screen?
Dr. Yes, now it's okay. Yeah. Feel free
Okay,
to go ahead.
thank you so much. For coming to a webinar on a late Friday afternoon, you must be very motivated statisticians wanting to learn something about handling missing data and how is it related with estimands. So today, I will give a quick overview and strategies for handling missing data. And particularly, the missing data issues have been exemplified during COVID-19 when missing data become a critical issue in the clinical trials. So I want to acknowledge the AVIS Missing Data Statistic Excellent Working Group, which me and Natalia was co-leading this working group for, I have been with this working group for the past several years. And we have a lot of discussions and works accomplished on, strategies for handling missing data. I don't claim to be an expert on missing data methodology, so this is really not a technical presentation, but maybe an overview to give you a quick overview on the strategies. So I would do a quick introduction, and then including regulatory guidance and impacts of missing data and impacts on data collections, and then going into overview and methodology for handling missing data. The way I organizing it is I organizing it by the type of variables that you might encounter. For example, continuous endpoints, binary endpoints, count endpoints, and time to event endpoints. What things you need to consider when you have missing data and methodologies to apply for it. And then most of these were under missing at random assumptions. And then I'll just touch a little bit on, some of the analysis that we often use as a sensitivity analysis for handling missing data under missing not at random assumptions. And then how is missing data versus the estimand? Particularly now the Estimand and ICH E9 addendum A1 is out for several years now. Okay. So a quick introduction. Avoid missing data from the beginning. I mean, it's very easy to say, but as a clinical trial statisticians, we do need to pay attention from the beginning. From a well-written protocol with a well-designed, to minimize the amount of missing data. For example, the choice of population, right? The number of study visits. We have one protocol, for example, we were negotiating with clinical and in number of visits, they wanted to have so many visits, it potentially become cumbersome for patients. And that we know is prone for missing data. And site selection. Some sites are better on follow-up, some sites are not so good. So we need to make sure the site selection part of it, even though it sounds like this is a clean ops job, it's not statistician's job, but we need to pay attention to those, because if missing data occurred during the study because of poor protocol or poor site selections, in the end, it becomes a statistician's problem, right? And also like dosing regimens, oral dose versus subcu doses, some doses, can easily, patient can take the drug home to use versus have to come in the office, doctor office, for injections. Study procedures to reduce patients' burdens. And another thing that I wanted to point out is like rescue medications. Rescue medication use often, particularly in the framework of Estimand, can become an intercurrent event or not. And that could also introduce some of the missing data issues, okay? And making efforts to collect all the data, allows protocol to specify data collection after study participants discontinue treatment can remain in the studies. So this is also, I would talk about it in the Estimand settings. How is it going to help? Create a comprehensive ECRFs, review them carefully, and thinking with ends in mind, like, if you're going to analyze this data, how is your CRF to be designed so that that all the reason for missings, data are being collected. Now, COVID-19 have give us a surprise, right? It's a unforeseen major crisis. And during that period of time, I'm sure some of you have experienced panic periods, where we don't have case report forms, for example, to collect major unforeseen crisis reasons for missings, for dropping out, or not being able to come to visits, So that's one of the things that you just have to keep in mind. Now, COVID-19 is one unforeseen major event. Is, for example, a war, break out in certain area where you're conducting clinical trial. And that's also can be an unforeseen major crisis, or a hurricane hit an island or hit Miami, Florida, unexpectedly when the site just completely shut down. So these are the things just to keep in mind even before the trial start to avoid missing data. And assess the impact on the analysis. When you do sample size calculation, do you have to factor in certain dropouts? Do you replace subjects who drop out? These are the things that you have to think about when you do the analysis. With the end, again, with the end in mind as a statistician. Missing data can come from all kinds of resources, and I've touched upon some of them. You can miss visits, procedures, treatment discontinuations, interruptions, loss to follow up. Yeah, we always say anything can happen, will happen in your trial. So you have to like really pay attention to that. Other missing data can caused by human error, for example, data entry errors. So then you cannot use that data entries, or transcribing errors, or even some technical errors like machine breakdown. So your lab data, are all bad. You can't use it. Malfunction of the machines. All that situation. So just keep all that in mind. So we as a statistician, we know that not all missing are the same. Not all missing are equal, right? So there's missing data patterns, you have intermediate dropout patterns. For example, a patient may just skip one visit but still come back. So you have intermittent missing, or monotone missing is meaning that they lost the follow-up, or after that, after a certain point, all the data are missing. Or missing mechanism. I'm not going to go into all these details. You know that, you have missing at random, or missing not at random, or missing completely at random. And therefore, in the analysis, you have to keep in mind that, the reason for missing, can it help you think about what type of potential missing mechanism it will be. Missing data, why does it matter? Of course, we know that to ensure the research and the findings to have a good quality, integrities, reliabilities, credibilities, validities, all these things is the original objective of conducting a clinical trial. You want your clinical trial to have all these qualities to make a claim. Oftentimes, we also have to adhere and comply with the regulatory requirements and the GCPs, GMPs, and all that stuff. So missing data does matters. Later on, I will touch a little bit about Estimand. Of course, with Estimand being introduced, we have 5 attributes: treatment population, variable, which is endpoint of interest, and intercurrent event, which is an event that happened after trial have been conducted, and then population level summaries. And then Estimand have 5 intercurrent event strategies, treatment policies, composite, hypothetical, while on treatment principle stratum, I'm sure you have all heard about this, over these years after the intercurrent event things have been introduced several years ago. And how is intercurrent event versus missing data? What's the relationship between them? I will quickly later on give a few examples. For example, how, which one's missing data, which one's really is taken care of by intercurrent events. Okay, guidance document. What we do, particularly in the pharma section, clinical trials, they are bounded by the regulatory requirements, and therefore EMAs, FDAs, and the global regulators have guidance documents, to talk about these things, like missing data, why they're important. Like this one came out several years ago, prevention and treatment of missing data in clinical trials. And also FDA has guidance of industry, on clinical trial principles, guidance on missing data in confirmatory trial by EMA. And then this ICH E9R1 addendum talking about Estimands. So just a few guidance documents for you to pay attention to. Then I'm going to go into the details on methods for handling missing data under the MNAR assumption. First, we'll talk about if your endpoint is continuous endpoints. For example, certain methodologies used to handle for continuous endpoint. Continuous endpoint is probably one of the most popular endpoints that we use in the clinical trial, right? And then some points to consider for that. Continuous endpoint, you can have repeated continuous endpoint or a single measurement post-treatment. Repeated measure means, for example, change based on weight. You actually measure weight on each visits. So you have multiple weight measurements post-baseline. But for example, for immunology, ulcerative colitis, for example, it's measured by endoscopy. Oftentimes, we only have one endoscopy because of the procedure and the expensive procedure. We have one endoscopy at baseline and then one endoscopy at the final visit. And so you have only one single measurement post-treatment. Most common methods for under MNAR assumption for continuous endpoint for repeated measure oftentimes is MMRM, which we all know what they are. And then another method is multiple imputation and a few other likelihood-based approaches. And for example, if you have a major unforeseen crisis at some sites or in your clinical trials, if your primary analysis is MMRM or MI, particularly like for us during the COVID-19s, we, what do you do? Do you need to change methodology or switch methodology? So MMIM method is a likelihood-based approach that it uses all the observed data collected from subjects to predict the mean of the longitudinal outcomes. It has a lot of theoretical advantages over the last observation carry forward or observed cases. This is probably 15, if I've been in industry for long enough, this is probably like 15 years ago. We were still using last observation carry forward when I was working in neuroscience or in some other places where we're using just the observed cases at a particular visit. These later on, we abandoned them. We don't use them anymore. I remember going to FDA, trying to convince FDA that we can change to MMIM. And FDA was skeptical at the time, saying that in your MMIM you have to specify all your variant covariance structures and all that stuff, that your outcome may not be unique, and we have to go back and forth. But now I think it's completely acceptable when you have a repeated measure, which is you use MMRM. So MMRM is valid under missing at random or missing completely at random, as long as the ignorable missing mechanism assumption is valid. Even though under MMRM framework, the missing data imputation is not explicitly applied because it's used all the available measurements for the estimation based on the maximum likelihood principle. But I always say it's inexplicitly estimate the missing data because of the inference it's using. So in a way, it's valid. You have to make sure the assumption missing at random is kind of valid. General considerations when you use MMIM, make sure that you include variables with minimum missings as a covariates. Because MMIM do exclude observations with missing covariates. If you have that covariates you have that as a covariate, so missing will be excluded. So make sure you pick a variable with minimum missing value as a covariates. Based on the simulations and the literatures, unstructured covariance structure is recommended. unless you have very strong evidence that a particular covariance structure is applicable. And if you have that justification, I would highly recommend that you pre-specify that covariance structures in the SAPs and provide the justification for it. Otherwise, we recommend to use unstructured covariance structure. Kenworth-Roger method is recommended. It's implemented in SAS using DDFM equal KR. Because compared to other methods, KR methods is more robust to the competitive covariance structure, or if you have a smaller sample size or imbalance between groups. And so we would suggest that you can use that method it's the SAS using it as computing the denominator degree of freedom for the test of the fixed effects. This is like I include a few typical SAS codes that you can apply it, like here, if you use KR, specify DDFM equal KR. And if you using the repeated measure using unstructured covariance structure, you can say type equal UN. Now, during the COVID-19, and like I said, it can be any other major unforeseen crisis. If you get whole bunch missing data because of that, and oftentimes we justify during the COVID-19 that most of these missings are missing at randoms. But if you, all of a sudden you got a whole bunch of missings from that, and you already pre-specify your primary analysis is MMRM, a natural question is, do I need to change my methodology? Should I switch to some other methodology that also valid under MI? We have done some literature searches and simulations. We found out that if missing due to unforeseen crisis is not severe, it's just moderate or mild, less than 20-30%, you want to continue to use MMRM rather than switching to MI, because MI have no added value compared to MMRM based on the literature review and also on the simulations. Particularly the standard error from MI usually are typically larger than MMRM. MI is more conservative in preserving type 1 error and less powerful compared to MMRM. These findings are consistent across literatures that we have reviewed. Now, if the missing data from this particular major crisis or become more severe, then you do have to discuss and consult either with the regulators or switch to MI for the primary analysis, or adding another MI as a sensitivity analysis. And re-estimate sample sizes if you really think that the missings from this type of missing is becoming a more severe issues. And there is a paper by Lu to estimate sample size when it's MMRM. It's using within subject correlation matrix, between visits, and then the retention rate from visit to visits, and it incorporated that and can give you a power assessment and sample size re-estimation. It has a website you can actually use that. Sometimes even for sample size planning, if you do expect missing more severe towards the end of the study, or at certain point, you can use this method to check, because oftentimes we just use change from baseline to the final visit, and not including anything in between when we use MMRM. It may, you can actually use this method just to double check your sample size. It's a good way to verify the calculations or thinking ahead of the trial. If you use MI methods for your imputation, which is also valid under missing at random, the MI procedure is not intended to estimate each individual missing data points. It's for each imputation sets. It draw a random sample from the missing value from the underlying distributions, where the probability of missingness of a variable only depend on the observed data, and therefore it's valid under MAR assumption. I want to point it out that because it's a simulation and sampling technique, therefore, MI estimators and variance are subject to random error. So this is, later on I'll point it out, this is why you have to make sure you specify the seed that you use so that the result can be reproducible. And we all know if you have implement MI before, there's 3 steps in implementing MI, right? The first one is you specify an imputation model, and therefore you generate M complete data sets based on the imputation model using the observed data. And then with the M completed data sets, you analyze each set using your standard analysis methods that apply to non-missing data set. And then the inference part of it is you synthesize the estimate and variance in step 2 from the M data sets produced using Rubin's rule, and come up with a one single estimate and variance. There are a few things to consider when you using MI for your continuous endpoints. In the imputation model, to include covariates that predict of missingness and or predictable outcome in the imputation model, and pre-specify them in your protocol or SAP. The variables should be, this is one thing that we noticed, that you want to list your variables according to the number of missing observations, from the most completed to the least completed. Somehow, if you switch the order, it will give you slightly different results and outcomes. So you want to list the variables in the order from most complete to the least complete, from less missing to the more missing. And when you apply MI, you might also want to think in your imputation model, depending on your situation, do you want do the MI imputation using all subjects, or separate by subgroups? Because depending on the situations, your some subgroup, if they somehow have a different patterns of missings and stuff, you may want to consider, impute within each subgroups. And this is very situational, so you really have to have a good justification and assessment on it. Another thing about the MI for the analysis is that pre-specify your number of imputations in your protocol SAP. Again, like I said, make sure you pre-specify the seed numbers so that it can be reproducible, because it's a sampling technique is subject to the variance. So you want to make sure that the results, when you submit it to the agencies or later on, it can be reproducible. Because if you use different seeds, it may give you slightly different results. The number of imputations recommended is greater or equal to 30. I used to do just 10 or 20, which give you sufficient efficiencies. But later on, when we review some literatures and papers, it basically say a rule of thumb is that the number of imputations should be at least equal to the percent of cases where with missing data. So that's something to keep in mind. I would recommend use 30 or 50, 30 to 50. Imputation algorithm. There's when you implement MI, there are choices in the SAS where you can specify data. If your data is non-monotone missing, you wanted to use MCMC and impute the missing data, and then using monotone methods. You can also, if you have a large portion of missing data, FCS methods can also be used, which can handle non-monotone missingness. And increasing the number of imputation oftentimes can help you reduce the difference in the estimate from these different methods. Sometimes you use FCS methods, you come up with one outcome, and then you use MCMC, or you may get slightly different results. But if you increase the imputation from 30 to 50, for example, the two results become less different. I want to quickly touch upon the imputation and efficiency. This is coming from Rubin's paper book. Basically, your number of imputation, when it gets to infinity, it produces an unbiased estimate and correct variance. But we know we can't do M to infinity, right? And so it calculates a so-called fractional missing information, and how it related with number of imputations you need. So as you can see from the table, when you have 30 imputations, it's pretty much quite efficient. It's already, if you have 20%, it's already reaching 99% efficiencies. So you can continue to increase it. I mean, these days it's probably not so important anymore in terms of computation times. Many years ago, when the computer's not as fast, the computation times and efficiencies are more relevant. Now, 99.7, 99.1, going from 30 to 200, it's probably doesn't waste that much of a time, to be honest. Here are some of the SAS codes for implementing the like two-step approaches, and then later on one-step approaches. I'm going to skip the SAS code because I have other topics. Now in the when you have like major unforeseen crisis causing a lot of missing data, our recommendation is if you pre-specify MI as an approach, you can continue to use MI. You don't need to switch to other methods. And also make sure that you follow the MI methods, and also generally the MI methods it is appropriate unless you have very specific reason to switch. Quickly touch upon if you only have a single post baseline measures, because if you have single post baseline measures, percent of missing will greatly decrease the power or point estimate of treatment effects. So you could then consider using MI for that. Otherwise, oftentimes people just use a simple t-test or a regular linear ANCOVA. Again, this is basically one of the key things when you have unexpected missing data coming from certain unforeseen major crisis, is that our main recommendation is that keep it consistent across projects. And that's a key. Like you don't want to, one study you switched to one methods and then the other study you don't. And in the event of single post-baseline measurements, you may want to do a power assessment and make sure that the your power is not greatly reduced by the missing data. Next, I'm going to go into the binary endpoints. Like I said, I'm organizing my talk by the type of endpoints. So for binary endpoints, we know that there are two types of binary endpoints, right? So what is binary? Binary endpoints is responders or non-responders, success or failed, remitters, non-remitters, right? And there's two types of binary endpoints. Most of the binary endpoints, for example, come from a continuous variable that were dichotomized based on a pre-specified threshold. So, for example, in the endometriosis studies, right? The pain improvement responder. So we define a responder. It's defined based on a threshold that we say, okay, if the pain score changed from baseline of improvement by 2 grades, then these subjects are a responder. So that 2 grade, that threshold, actually coming from some kind of ROC analysis that you need to psychometric validate it and then get FDA alignment. So those are endpoints that are dichotomized from a continuous measurements. Or, for example, like Crohn's disease studies in a immunologies is defined by a score less than 150. Crohn's disease activity index. And where's 150 come from? Actually, we search literature. We find one literature that many years ago, somehow they did some kind of a cutoff of 150. So now it become a kind of a acceptable standard, cutoff threshold to cut off. So that defined the remitters. Some endpoints are just binary endpoints are not dichotomized from a continuous measurement. For example, in the HCV studies, the SVR, which is sustained virology response at week 12, it's a binary variable that's by nature. It's defined as the patients who has undetectable viral load 12 weeks after the treatment has ended. So it just, if the viral load is undetected, they are a responder. So it's a binary endpoint by nature. It's not defined, cut off from some kind of variables. For binary endpoint, you also have repeated binary endpoints. You can CDAI your measure. So if your variable is repeated measure and your binary endpoint derived from that continuous variable, so then you have a repeated binary endpoint. Or a single binary endpoint, same thing here, the example of ulcerative colitis. If you only do endoscopy at the final visit and you define your remission is reduction in some kind of measurement by 30%, then you only have a single binary endpoint at the end. And so the methodology for it's very similar. So you can use MMRM for binary endpoints or use MI for binary endpoints. Now, interestingly, for binary endpoints, there's one method called non-responder imputation, NRI, are often used. Now, NRI is valid under MCAR. It's actually not valid under MAR. I always hear people saying, oh yeah, NRI is a more conservative approach. But that is actually not true. It's not necessarily a conservative approach. For example, if you study a subject have more dropout, for example, in the placebo group, and you use non-responder imputation, which you impute all the dropout as a non-responder, it's actually discriminating the placebo. So it's technically not a conservative approach. It's often required by regulatory agencies. They just say, okay, you have to use NRI for your binary endpoint. If your responder definition is by a certain variable and that variable is missing, the subject is a non-responder. It sounds like more conservative, but it's not. Another thing I wanted to point it out is that the non-responder imputations, particularly you have to be careful when you're doing non-inferiority trial. We have recently a non-inferiority trial and we use NRI, and actually we have agency come back and say, because it's non-inferiority trial and you have dropouts, we want you to use MI for certain missing data and NRI only for other certain missing data. Because the non-responder, the non-inferiority trial, you're trying to detect a lower bound. And if your dropout kind of not the same, or even the same between the groups, NRI actually may favor an inferior drug. For binary endpoint that are dichotomized from continuous variable, when you're doing multiple imputations, for example, there's again two ways to do it. One is you can impute the continuous outcome before you dichotomize the response, or you dichotomize the response and then impute the missing binary response. And we compare these two, the literature also compare these two. The recommendation is to use IBD, is impute the missing continuous outcome before dichotomizing into the response. Same thing for major unforeseen crisis. If your original analysis is non-responder imputations, you could argue that because the missing from the major unforeseen crisis is missing at random, therefore those missingness you can use multiple imputation, and then therefore you can have a hybrid methodology like NRI for all the other missing data, and then multiple imputation for missing due to COVID-19, for example, or due to natural disaster. So here is just a quick illustration of applying continuous variable imputed first, and then override it and dichotomizing it during the last stage and not in the imputation stage. And then do the analysis and the inference stage. And in the imputation step, you would impute the continuous variable itself, not the response. For variables that are not derived from a continuous variable, there's in the SAS, you can have different ways of doing it. You can apply monotone logistic regression, or FCS logistic regression, or MCMC multivariate normal, which assume multivariate normal. I guess the recommendation is using the FCS logistic is recommended due to it can obtain the more unbiased and robust estimate. And then it performs better than any of these discrimination functions or MCMC multivariate normal, where you have more assumptions into it, and it's better than NRI and observed case too. So we would recommend using FCS logistic when your binary endpoint is not dichotomized from a continuous variable. So you only have binary variable itself. So you use FCS in your imputation. Pretty much same thing for unforeseen crisis. If it's not derived from a continuous variable, you can use MI FCS logistic, or use MI monotone logistic if it's close to monotone missing data. For the rest of the missing data, you can use your whatever the method you pre-specified. I'll skip the SAS code. This SAS code gives an example how to use FCS or monotone methods in the logistic regression model. Count variable. So count variable oftentimes includes the count, which is not binary, not 0, 1, responder or non-responder, but for example, number of asthma exacerbations. It's 1, 2, 3, 4, 5, 6, 7, or episode of certain side effects. You have 1 episode, 2 episode, 3 episode, 4 episode. The methodology for count endpoint involved often is a Poisson regression or negative binomial regression. And in case of excessive 0 in the observed data, oftentimes, there's no event, and then like 3 exacerbation. So you want to use some kind of zero inflated count model to apply to this kind of situations. Things you have to pay attention for a count model. If subject drop out, only the observed count before dropout should be included in the analysis. None of these methods handle missingness in the covariates unless you make them a separate category. So for Poisson and negative binomial regressions, oftentimes the response are not normally distributed, and the variance are likely to increase with the mean. And this dispersion issue you have to take into considerations. Another reason that can cause dispersion is the heterogeneity due to the ignorance of the importance covariates in the model. So negative binomial is a common way alternative to Poisson regression, which can address a little bit of the over dispersion issues by including a dispersion parameter to account for the unobserved heterogeneity. So if you think your subject heterogeneity is slightly more severe because of, for example, site selection, it's just all over the places, you can use negative binomial regression instead of Poisson regression. Same thing, we have some SAS code that you can use the Glimix or GEMMOD. Time to event endpoints. To be honest, I am least familiar with time to event endpoints because I have actually not working in, like, it's very common in the oncology therapeutic area, right? Which is a therapeutic area that I have not worked with. So so methodologies oftentimes, if you have missing data, it's considered, if it's missing at random, for example, the data you think is missing at random, then it can be considered, you can use non-informative censorings. And therefore, if it's missing at random, then the standard survival methods can still be applied. The death event somehow is one category that you have to, it's in the next. Yeah, death is one thing that you have to be careful if, for example, like during the COVID-19, there's excessive death that happened due to the unforeseen crisis. For example, war in certain area causing somebody got gunshot and dead. And that actually becoming a competing risk. And so you have to take that into consideration rather than say, oh, it's missing at random. And therefore, I'm just going to use the standard survival methods. So in that case, you want to make sure you take a competitive risk factor into consideration and consider some kind of different type of analysis as a sensitivity analysis. That's all I can say for you that you need to pay attention to. The rest of the talk, I will touch upon the sensitivity analysis under missing not at random or MCAR assumption. For missing not at random, my experience, I have used two most popular ones. One is called pattern mixture model. You defined the patterns. You define the missing data into different patterns where you justify the number of patterns. And oftentimes, if you do apply this methodology, for example, you wanted to pre-specify your patterns in the model, like how many patterns you want to group your potential missing data into. Like when we do COVID-19, we treat COVID-19 as a separate patterns from the others. Another very popular methods and oftentimes being required by FDA these days is tipping point analysis. You know, so what you do is missing data are replaced by a range of values to observe how far the shift is needed in order for the study results to tip over to, for example, from significant to non-significant. For example, we were used by FDA and saying, well, in our Crohn's disease studies, FDA say, well, we don't know if your missing data is missing at random or not. And we like you to add a sensitivity analysis, to handle missing not at random. So we proposed PMM analysis, and that was accepted as a sensitivity analysis. Another study is we add a tipping point analysis to primary and ranked key secondary endpoint in another immunology studies, and it was requested by FDA, and so we did the we did add that as a sensitivity analysis. Another example recently we got from FDA on Bayesian borrowing. I want to share this with you. Although it's not a tipping point per se, what we were asked to do is that we have a pediatric study and we want to borrow information from adult. So FDA say you can do a weighted borrowing, like, you have W times a non-informative prior times 1 minus W times your adult distribution. So it's kind of a hybrid weighted from non-informative to informative prior. And you vary that W from 0 to 1 by like 0 0.05, 0 0.1, 0.2, and to see which point, how much you borrow, for example, the results. So I kind of view that as a tipping point analysis, even though it's not the typical and traditional tipping point analysis. So for missing not at random, oftentimes we assume it to be you can group certain reason as not missing not at random, but then you can group some as a missing at random. So, but you can define your patterns that way. We also noticed that missing not at random imputation methods usually provide more conservative estimates than missing at random methods. For example, the order of conservative would jump to reference to CRs to CIRs. So even within the missing not at random methods, the results and conservativeness can be different. I mentioned that you have to define the missing patterns ideally, if you can pre-specify your missing pattern, will be better. So for tipping point analysis, usually it's better to perform at the subject level and not at the population level. I'll skip the MCAR because we don't normally use it like completer case imputations or a single replacement or substitute replacement for missing value. For example, use mean of a variable to replace the missing for that particular variable. We don't do that anymore, so I'm going to skip that. Missing data versus Estimand. This is an important topic because given the Estimand framework, we know the most important thing is that we need to think about during the design stage, what is your target of estimation that reflects your objective, right? And there are the 5 attributes of the Estimand. And the most important thing I highlighted here is the handling of intercurrent events. All the other ones we know it's important, but the handling of intercurrent event really is linked kind of with the missing data. There are 5 strategies. We all know that. I'm going to skip that. So ICH E9 make a differentiation between the missing data versus sensitivity analysis versus supplemental analysis. You all know that sensitivity analysis is you use different methodologies on the same Estimand. For example, that's sensitivity analysis. But if you change any of the attributes, all the methodology, all the strategies for handling intercurrent event, that change Estimand, and therefore it's a different Estimand, and they call it supplemental analysis. Missing versus intercurrent event. In the composite variable strategy, for example, if you use NRI, then subject who drop out of study, for example, is defined as and therefore data after that is really not missing. I mean, because you already incorporate the missingness into the definition of the variable. So by using composite strategy, you don't, like subject drop out of the study is really not a missing data anymore. That's how I see it. For example, another example is if you use one on treatment strategy, any data after the subject discontinued the study treatment is technically not considered missing because you only by specifying one on treatment strategy, you only use the data while they are on treatment. So anything not collected after the treatment discontinuation is really not missing, So, but any of these, you need to be pre-specified during your protocols, how you do that. Okay, I think this is all I have. I really appreciate you for staying and going through the talk. As I said, the job for statistician in pharmaceutical is easy if everything is as theoretically as planned. If you have a complete data set, if your p-value is less than 0.001, your job is easy. However, it's never that way. You will have encounter missing data, different type of missing data, and your p-value will be borderline, and then you will be asked to do tipping points when the result will tip over and become not significant. And so these are why the statisticians need to be on top of all these methods and know that when you need it. Okay, thank you.
Okay. Thank you very much, Bidan, for the comprehensive talk on this missing data thing. We do have, I think we are over time. I
Okay.
think we do have, I think, 8 questions. So maybe we can go quickly answer the questions.
Yeah, Dr. Biwan, are you able to see the questions in the Q&A? If not, I can copy paste it in the chat.
Yes, I can see. Yeah.
Okay.
Yeah, let me. The first 2 questions is related to the mentor program. Maybe I just read the answer. We get the answer here. The first is does one need to be in RCSA member to join the mentor program? You do not need to be a RCSA member to join a mentor as a mentor, but you do need to be RCSA member to be a mentee. This is the first question. The second question is the mentor program free to join as a mentee? Yes, at least for students, RCSA membership is required for mentee, but RCSA membership is free for students. So this is for the mentor program. Let's go to the missing data questions. We have answered 6 questions. Maybe I'll read one to one. You can answer.
Yeah.
First is you saw that first one is there a rule of thumb saying whether total missing data?
Yeah.
So we are not. Yeah. 5%.
Yeah, I have noticed that through all the simulations, if missing is less than 10%, any of these methodologies for handling missing data pretty much give you a very consistent results and outcomes. When the data between 10 to 20%, that's when I call it moderate. Then you have to pay a little bit attention. When the missing is more than 30%, that's when I would call it more severe missingness. So then you have to be extremely careful about your missing imputation models.
Okay.
I would say I would use 30 as a severe as a cutoff.
Okay, thank you. So second question, Bidan, for MNI, can MMRM be used as an imputation model?
Yes,
Okay, thank you.
definitely.
Okay, next question. In case it's not discussed, I would like to opinion the following. Okay, maybe next question. Control-based imputation approach in pattern mixer imputation for the treated arm is usually considered as a conservative in a non-treatment controlled trial. However, it is not necessarily the case for a non-inferiority trial with active.
I agree.
You agree?
Yeah.
Okay.
I agree that comment because non-inferiority trial is actually opposite because you think your drug actually is inferior to the drug that you're comparing to. And so kind of the tide is turned around. And so you have to be careful that in terms of conservativeness versus non-conservativeness, oftentimes we think conservativeness in the case of a superiority trial, but in the non-inferiority, it could be opposite.
Okay, thank you, Bidan. Next question. Is there a percentage remission data threshold or range?
I answered
MNAR
that.
or other methods does not provide reliable estimates anymore? For example, 20%, 30%, or others?
Yeah, I answered that. My rule of thumb is 30% become more severe. You really have to then evaluate the missing reasons and use different methodologies to check for consistency. If it's less than 10%, our simulation show most of the missing methodology give you a pretty consistent outcome.
Okay. Do you have any reference I recommend Bidan? For this one, for this MI percentage missing threshold?
I'll check to see if any of them if I have a paper that's referenced that. Yeah.
Okay. Okay. Sure. Okay. Thank you. Next question is quite long. IBD approach for binary endpoint, how to handle in the current event using composite strategy. For example, if there's in the current event, one subject is a non-responder by a composite strategy, how to impute the underlying continuous value. If the continuous value is imputed, this will use the hypothetical strategy, assume missing after the increment instead of composite strategy. Could you help clarify?
This is a very good question. I mean, you can have a different ways of doing it, right? You can say, for subject who, is a composite by certain value, you can impute the value and then just set them as a non-responder regardless of what the imputed value is. In certain category of patients. For example, if all the discontinuation of, or if all the subject using, I would still use IBD, but you impute all the values, then these group of patients who, for example, you use non-responder, you would manually set them to non-responder. And then in your analysis, they become non-responder rather than using the threshold to cut them as a non-responder.
Okay.
So it would involve a manual step to, in the imputation step, you can impute value for these subjects, but there is a manual step involved where these group of subjects is non-responder. You will artificially set them to be non-responder before you do the analysis for the complete set, before you apply the threshold.
Okay. Thank you, Bida. The last question asking acceptance of tipping point analysis by FDA. Do you have any reference for that?
We have actually, I don't have reference, but we have getting like multiple projects requested by FDA to do tipping point analysis. So I assume that it's accepted by FDA. And actually, we have several programs where we propose tipping point analysis to FDA as a sensitivity analysis. So I want to make it clear, it's sensitivity analysis, not primary analysis, and has been accepted by FDA.
Okay. Yes. Thank you very much, Bida. Answer all those questions. I think it's a wonderful talk and generate a lot of interest from the audience. Maybe I think it's time to close today's presentation. Thank you very much again, and
thank you so much for staying online. I can't see the note for the time reminder, so sorry for overtime. Thank you so much for taking Friday for the talk. Have a nice weekend.
Yeah,
Okay,
thank you, Dr. Deli and Dr. Buda. I will follow up with you guys in email. Yeah, okay. Thank you, everyone, for coming. I will close the webinar now. Okay, bye-bye.
Bye.